Hi everyone, welcome to the sixth of our HPC Perspective Live. My name is Natalia Jimenez and I'm the Life Sciences Industry Lead at Viridien. I am delighted to be your host today as we continue to explore HPC topics. In this series, we have been traditionally introducing you to a number of people across our group who share with you different aspects of our AI, HPC, and cloud solution business at Viridien. However, today is different as we have the pleasure to speak with Dr. Gemma Holliday, Associate Principal Cheminformatician at AstraZeneca. Gemma will share with us her thoughts about the challenges that biopharma companies are facing and the opportunity that artificial intelligence can bring to tackle them. Gemma has a background on chemistry from the University of St. Andrews and has worked at the interface of chemistry and biology for most of her career. After working at the European Bioinformatics Institute, she moves to University of California, San Francisco as executive director of the structure function linkage database and then joined Medicine Discovery Catapult as a bioinformatics data scientist in 2017 and running the chemoinformatics team. She joined AstraZeneca a year ago as Associate Principal Chemoinformatician. Gemma, welcome and thanks for joining me on the virtual couch.
Thank you very much. It's a pleasure to be here.
Thank you. Gemma, you have an impressive background. Can you tell us about the achievement you are proud of?
Absolutely. I think data are at the forefront of everything we do. And I've had amazing opportunities in my career to work with some fantastic figures, from Professor Dame Janet Thornton at the EBI, the European Bioinformatics Institute, to Patsy Babbitt at UCSF. And that's really helped shape me as a scientist. But it's really challenging to point to one thing that I'm most proud of. But I think probably if I had to say one thing, it would be my work with the Macy database, which we rebranded a few years ago as the MCSA. So Macy stood for Mechanism, Annotation and Classification in Enzymes, and the MCSA is the Mechanism and Catalytic Site Atlas. And I think that's definitely top of my list. So what why is that? It's because we know so little about biological space in which we operate and in which life operates. If you look at the statistics, if you just look at the raw numbers from something like UniProt, which is one of the flagship sequence databases, it's actually a little scary how much we don't know, but it's also an amazing opportunity because there's little over sort of 570,000 proteins and their annotations that have actually been checked by a human. Um, which sounds like quite a lot until you think about the fact that there's over 248 million as of May this year protein sequences that have been identified. And that number is growing almost exponentially. There's a huge amount of opportunity here. And we rely on the data that's available for us to be able to predict the function of a protein or the likelihood of an enzyme to catalyze a non-biological reaction, amongst other things. So resources such as the MCSA are really critical to giving us a good grounding from springboard into the unknown. I started Macy in 2002 during my PhD. Over 20 years later, we're finally in a state where we can utilize the data to make predictions on catalytic mechanisms. That's partly due to advances in data science and computational techniques over the last 2 decades, which really have been immense. I mean, has anybody heard of ChatGPT? But it's also because it takes a while to build the level of knowledge we need. The distance between 2 points will always be a straight line. You need significantly more than 2 points to create the Mona Lisa.
I'm incredibly impressed by your achievement and the remarkable legacy you have established in the field of bioinformatics. Your work exemplifies the significant advancements that have been made in the industry. It's amazing to see how, over the years, pharmaceutical companies have been building their computational capabilities to shift workloads from the web laboratory to the computer. This area has become very broad and specialized, encompassing a wide range of expertise. In this rapidly evolving landscape, I'm curious to learn more about the role of a principal chemoinformatician and how it contributes to this advancement.
As you said, there are so many different ways we can use computational approaches to augment our workflows. And incredible number of different flavors of computational science too. We can go right away from the quantum mechanics experts to data scientists to informaticians, and every single one of us plays an important role in supporting what we do in the wet, to supporting the wet lab experts. Those people who actually do the physical experiments at a bench with real samples of stuff. Um, so I'm personally in the informatics camp. And I've spent most of my career at the interface between biology and chemistry, as you mentioned in the introduction. And so I kind of self-identify as both a chem and bioinformatician. But what does that actually really mean? So informaticians are data experts that use what's available to us to turn data into knowledge. And here I'm talking about data as discrete elements of information. So a melting point, an activity, a physical measurement, and knowledge is how we put that information together to make sense of it. So I often liken informatics to cooking. So we take all the data we can lay our hands on, we mix it all together, we stuff it in a computer, and we see what comes out the other end. Sometimes it's a gourmet meal that has the potential to revolutionize the way we work. Sometimes it's a limp souffle, especially if the data we use in the first place is of poor quality. I feel it's an important point, though. We can't do much computationally without the underlying data that is produced by our wet lab colleagues. Yeah, we can build a machine learning model on to predict the outcome of an experiment, but we need to be aware that these aren't always correct. An awful lot of effort goes into identifying the accuracy of models before we use them in anger. We rely not only on positive results, but those negative or it didn't work results, because if we don't use those, our predictions, we really need to use those to ensure that our predictions are as accurate as possible. If you only train a model on positive results, then you'll only ever get yes as the answer. And yes isn't always the answer, unfortunately. And our toolkit is evolving at, frankly, an incredible rate. With the advent of large language models, the LLMs, and more powerful computers. I mean, quantum computing is edging closer all the time. These advances really bring what was once a domain of experts to what we might call the average user, and gives us ever more complex and powerful predictive tools, which will allow us to go further and faster than we have before, but with a warning. If you put garbage in, you will get garbage out. Everything we rely on, everything we do relies on good data, quality, structured, balanced, unbiased, and ethically fair. Absolutely everything.
Yeah, definitely. Data plays a key role in the drug development process, which, as we all know, is lengthy, complex, costly, and with a very low success rate. Could you walk us through a typical drug discovery pipeline? I would like our audience to better understand how intricate and challenging this process is.
Absolutely, as there are many different ways of starting the drug discovery but the most common one is to start with a disease or a protein target, which we tend to call target identification. That sounds simple, but it's actually really complex because biology is incredibly complex, and Mother Nature has a surprising ability to find exceptions to almost every rule you could think of. We now understand better that proteins don't exist in isolation. They're part of a complex ecosystem of processes and metabolic cascades, and they can exist as well in different parts of the body, sometimes with radically different functions. Maybe it was easier when we knew less. But knowledge moves on, and then once we have the target, we look for substances that might be active against that target. This is a process we call hit identification. Historically, these substances were almost exclusively small molecules, but nowadays we have a huge range of different modalities that people have developed, from small molecules themselves, which are still critical molecules, to antibody drug conjugates, which are small molecules that are tacked onto the side of an antibody, cell therapies, to gene editing. However, the idea remains the same. We find something that will modify the activity of the target. Once we've got that list of putative substances, we can refine, which we call lead generation, and improve the candidates, which we call lead optimization. This is the essence of discovery. Once we've done that, especially in the case of small molecules, we'll go from a chemical space that is immense, especially if we include generative techniques, to a couple of leads at this stage, and we typically will create milligrams of substance. That's when the fun starts. We then need to ensure the candidate that we've chosen is stable and safe. We need to figure out how to manufacture it at scale. We need significantly more than a couple of milligrams for clinical trials and commercial applications. We need to figure out what makes the best formulation and the dose at which we need to administer it and how we're going to administer it. Is it a tablet? Is it an injection? Once it's passed the various safety and efficacy checks, we can go into clinical trials, which explore whether the drug is actually going to make a difference to real people with the disease being targeted. Finally, we go through a submission process where the various medical and regulatory authorities, who ensure we've done everything possible to meet the safety and efficacy criteria, even after we've got approval and it's being given to patients, we never actually stop collating data. What adverse effects are being reported. We check every single batch against the specifications originally defined, and occasionally refine those specifications because we monitor the sales and the usage. We never really stop collating data. And this process isn't always as straightforward as I've described. We all do our best to ensure we put patients first.
Oh, wow. What a journey. Thank you very much for this comprehensive overview of a process. Now, could you highlight some of the challenges associated with drug discovery in the pharmaceutical industry?
So the thing I've probably not spoken much about yet is the social and environmental impact and challenges of our industry. Of course, our main goal is to treat people with medical conditions, but we also have a duty of care towards the environment. Everything we do has an environmental impact, from our choice of solvents to the electricity we use to power up our laptops. Every new technology we utilize that has the capacity to revolutionize our ways of working also has a cost. For example, I recently read that training and using large language models has been predicted to consume as much energy as the entire country of Ireland over the course of a single year. It's a sobering thought. Of course, this doesn't mean that we shouldn't use these tools. It just means that we need to ensure we're using them responsibly. We also need to consider the impact of issues such as comorbidities, having more than one medical issue, which is much more common as we get older. An aging society and the buildup of resistance to drugs, including the rise of bacterial resistance to antibiotics, what happens when an infection doesn't respond to anything? I believe we all have some responsibility as an individual to do what we can to reduce our impact on the environment and operate sustainably. And we at AstraZeneca take that responsibility incredibly seriously as a company too. We want to ensure that our legacy isn't just a healthier population, but a better environment too. One of our goals is to be carbon negative across our value chain for all residual emissions by 2030. It's a bold ambition, but it's one that we believe in.
Given these challenges, how do you see artificial intelligence playing a role in addressing them?
So you might have picked up on the fact that I've not actually used the term artificial intelligence much so far. It's a bit of a personal foible, maybe, but to me it's a bit of a loaded term that for some may conjure images of Skynet and job loss. Of course, we do know that AI is extremely beneficial within our industry, and it's also a term that everybody uses, so I suppose I can't really escape it. Fundamentally, when we talk about AI, what we really mean is using computers to augment our ways of working and explore beyond what a human alone can manage. We are never going to manage to physically characterize all 248 million proteins, for example. It's just too much. This is, of course, an incredibly rapidly evolving field. I mean, almost every day you see something new coming out, especially around LLM, large language models. There are also lots of ways that these methods and tools can help us. Something we've been doing for many years is known as computer-aided drug discovery, where we use diverse techniques and methods to identify targets and hits, and then sort of improve those hits through lead optimization. There is a really diverse and exciting landscape of companies out there, including AstraZeneca, and academics who are pushing the boundaries of computational drug discovery. And it's becoming a standard approach in these early stages of the discovery pathway. We can also explore using digital twins, where we replace human and animal models with theoretical versions. We can build models to predict the properties of drugs that help us do more targeted experiments and reduce the burden of experimental wastage. Fundamentally, All these methods are about augmenting our ways of working, allowing us to do more with the resources available to us, alleviating the pressure on our scientists to give them more time and space to do the cool, challenging stuff. Humans have a huge advantage over computers. Critical thinking and creativity really are our superpowers. As I alluded to earlier, there is simply too much data out there for humans to realistically review and digest. And we aren't going to stop producing data. Yet with computers, we have the ability and the chance to utilize and drive innovation based off that data. But there is a caveat. The data we use to train our models can be biased. It can lead to inaccurate results. It can lead to dangerous results. There have been a number of incidents in the media recently around the pitfalls of AI, from biased recruiting processes to large language models inventing legal precedents. Good quality data sets, well structured and unbiased, at least as far as you can, are critical. But our data also needs to be fair and ethical. Whilst we need to be sensitive of how people identify, for example, gender, there might be conditions that are linked to the Y chromosome, somebody's biological sex. I don't want to go into too much detail here. I'm not an expert by any means on this, but ethics of data are really important. Maybe even more so when it comes to the data people have consented to us hold and utilize as part of our clinical trials. We are all biased, and it's worth remembering that large language models are trained on human input, so they can be biased as well. As long as we remember to trust but verify, there's hope. And as we continue to explore what's possible, we really do have the ability to change the world.
That sounds very promising. Can you provide some examples on how your company is leveraging AI in drug discovery?
Yeah, we use AI in lots of different ways. From early stage discovery, the number of possible drug like compounds greatly outnumbers the stars in the universe, to smart factories, interconnected, digitally enabled supply networks comprising our develop and manufacturing sites and our end to end supply chains, along with our external suppliers and partners. No matter what we're using it for, though, it's about helping deliver quality medicines to the right patients with the right safety and efficacy. We can also use neural networks to predict molecular properties. Is the compound going to be absorbed well? Can we get it to the right place in the body? How likely is it to be metabolized and eliminated from the body? Challenging questions, even with machine learning, but things that we can start to explore with existing methods. We also utilize knowledge graphs a great deal. These help us harness vast networks of scientific data to draw novel insights about genes, proteins, diseases, and drugs. More importantly, they elevate the relationships between these things, how they interact and how they work together or not, to a level that enables greater understanding on how these relationships can be utilized for drug discovery. I'm not covering everything by any stretch of the imagination here, but we can't forget the digitization journey we're all on. How do we ensure we're capturing our data in a fair manner? And here, I mean, by fair, I mean findable. Can we find the data? Can we access the data so it's accessible? Is it interoperable? Can we use it from different systems? And is it reusable? Can we actually reuse it? Um, and how do we empower our colleagues to use digital techniques to enhance their own workflows? Fundamentally, it's not just about the bespoke methods and tools that are built by experts, but about the democratization of digital techniques.
Fascinating. What do you envision for the future of AI in drug discovery, and how do you think it will shape the pharmaceutical industry?
Honestly, I don't think we've even seen half of what could be possible. If you'd asked me a few months ago, I'd probably been quite negative about things, especially the large language models and Chat GPT. But I've really had the opportunity recently to use Chat GPT, and I've and have been honestly pleasantly surprised. For the casual user, artificial intelligence will really start to impact in terms of taking some of the boring, repetitive tasks off our plates. Summarize this? Sure, no problem. As we get into more specialized use cases, we can start to see some of the power. A good example of the public domain in the public domain is AlphaFold. There's no way we could create the crystal structures of all the proteins in UniProt as quickly as AlphaFold has been able to predict them. Of course, it's not perfect. It doesn't always produce something that makes sense, but it's a heck of a lot closer than nothing. And we can now expand our computer-aided drug design methods to more sometimes very different proteins, as long as we use our common sense as well, trust but verify. Where will all this end up? Who knows? But I don't think we'll get to the stage in my lifetime when humans are redundant. Maybe we should rename artificial intelligence to augmented intelligence. All these methods really are about enhancing what we can do by exploring the data available to us. We still need to imagine what's possible and check what comes out the other side.
We are all aware of how quickly AI technology is evolving. How do the companies keep up with the pace of change? Do they have skills and expertise to embrace the full potential of AI?
It's really hard right now to keep up with the pace of things like ChatGPT. It's evolving so quickly, and we're only just scratching the surface of the legal and ethical implications of training a model on the internet. However, the beauty of things like ChatGPT is you don't actually need to be super technical to use it. You just need good old fashioned human curiosity, a dab of patience. Sometimes these things can get stuck in a rut or double down on errors, and the ability to think critically. Prompt engineering is possibly the most valuable skill you can build when it comes to ChatGPT. And AstraZeneca is actively encouraging people to upskill in generative AI, from learning how to write good prompts to verifying results and understanding the underlying biases. However, like I've said before, AI augments human intelligence. It's still reliant on us as humans. You still need to check the output. Is it correct? Or has it made up the answer? I recently asked it to create my biography for my CV, and I never knew I'd won so many awards. It's great for writing code, but you need to check it's not introduced security vulnerabilities and that it's using the most up to date version of the packages. Remember, it's trained on the internet and it's trained on the internet from yesterday, not today. Interestingly, the output also isn't very reproducible. I suppose it's a bit like asking humans. If you ask 5 different people the same question, you'll get 5 answers of varying levels of veracity and similarity, maybe more. I've not spoken much about the skills needed for the more hardcore, I suppose, methods. Machine learning models that require more technical expertise. These are things that the vast majority of people will never approach. But for these, many of the same skill sets are needed, just with a bit of added computer science, statistics, and data engineering. However, even here, AstraZeneca is helping to empower people by providing data science training for our wet lab scientists.
To finish with, there is lots of exciting developments in this space. Is there anything in particular that you are most excited about? Tell us about the most innovative initiative you've come across and the impact that it will have improving patients' outcomes.
Honestly, I think the biggest development is the field of generative AI. For example, with the advent of large language models such as ChatGPT, because because they're trained on so much data, it's remarkable how powerful they already are. A lot will come down to how we decide to utilize these, though, from simple tasks such as summarizing workshop outputs to spotting links between concepts. How we use it is somewhat limited by our own imaginations and the ability to write a prompt to produce the response that we're happy with. But humans definitely aren't optional.
Thanks, Gemma, for being with us today, and thanks everyone who has joined the live stream or the recording of this. If you got some questions for us, feel free to reach out to us individually.
And thank you for having me.
Thank you.